numbers of these 20. And they're linked together by peptide bonds. Another name for that would be an amide bond. And you link them together in a dehydration or condensation reaction. So here's two amino acids. Lysine, with its R group, is a hydrogen. In this case, alanine, its R group is a methyl group. So there's the central alpha carbon with its R group, hydrogen, amino function on one side, carboxylic acid on the other. It's going to condense with this amino acid, which has the same structure, only differs in the R group. The R groups don't participate in a polymerization reaction. It's just a linkage of the carboxyl group and the amino group. And we're going to take the elements of water away from these. So we're going to take a couple hydrogens off the amino group and one of the oxygens off the neighboring carboxylic acid to give us water. And the nitrogen and the carbon link directly together to make this peptide bond or amide bond. So it's a dehydration reaction to build up the polymer. At each step you do another linkage like that. So what do you suppose the reaction is called when you digest a protein and break it down to its amino acid? Chemically, what kind of reaction is that? Hydrolysis, yeah, we'll add the water back. So dehydration in the synthesis direction, hydrolysis in the breakdown reaction. So we now have what's called a dipeptide, two amino acids linked by peptide linkage. Okay, the dipeptide, notice it has we've only messed with the central groups. It still has one free amino group and one free carboxyl. There's one on this end, there's one on that carboxyl over here, and over here are not touched by this condensation reaction. Only these two, this amino and that carboxyl. So guess what? This is free. This carboxyl can react with another amino group from another from another amino acid. So we can grow a dipeptide into a tripeptide, a tri into a tetra, and so on. You can, in theory, there's no end to how long a protein chain can be. And as we talk about proteins, just as we talked about DNA having directionality, you're asking a question that have about 5' and 3' ends, you can identify directionality in growing peptide chains. Because no matter how many amino acids you link, it's going to have the same similarity to all of them. One end will have an underivatized amino group. We call that the N-terminal end or amino-terminal end. The opposite side is always going to have a free carboxylic acid. We call that the carboxyl and the C-terminal end. So there's a certain head-to-tail or directionality nature to a protein. This is the direction it grows in the cell, by the way, in the ribosome. You're always adding to the C-terminal. This one is the first one. It's usually methionine, and then things get added, going elongating on the C-terminal end.